In this lecture, I wanted to spend a little bit of time specifically on the light receptors for our special sense of sight, because these are kind of the trickiest of kind of all of the special sense receptors. In this lecture specifically, we are going to talk about a little bit about what light is, but also what the rods and the cones actually do for us. Those are the photoreceptors in our eyes. And we'll talk about how when these photoreceptors are activated, how that actually leads to our brain perceiving vision. So I just kind of want to go over the basics of what light is, how light is perceived, and how our brain kind of takes all of that messaging and creates sight. So first off, it's important to first just kind of ask ourselves our eyes, but then our brain. So light is actually a form of electromagnetic radiation. And because of that, so there are these little particles of light, which I'm just drawing as a little dot here. And those particles are called photons. Photons carry energy. And when photons travel, they specifically travel in waves, which we refer to from peak to peak as a wave length. Different colors of light have different wavelengths. So you can actually measure the distance between peak to peak, and that is your wavelength. And there are a lot of different types of electromagnetic radiation as shown by this figure here on the right. For instance, things like x-rays, which are used if you break a bone or when you go to the dentist once a year, they usually take an x-ray of your teeth. UV rays, those UV rays that give us that tan we always want in the summer, but also can give us that horrible sunburn, are a form of light, an electromagnetic radiation. And then we have the visible light spectrum, which I'll talk about here in a second. And we have things like microwaves, which cook our food, and radio waves, which allow us to play the radio when we're driving our car. But visible light, which we're going to focus on today with the sense of sight, there are basically the essential colors, which you can always remember by the acronym ROY G BIV: red, orange, yellow, green, blue, indigo, and violet. And these photons travel in a spectrum that is capable of being registered by our eyes. And so, again, we can measure the wavelengths of light, and that's what these distances in nanometers are telling you. So blue light travels at 420 nanometers. A nanometer is 1/1,000,000th of a meter, so it's really, really tiny. 9 zeros there, right? Was that a billion? Yes. And then we have kind of all of the visible spectrum, and you can see that we have certain receptors that pick up certain light forms, and those I've just starred there. The way light actually works is that these wavelengths, they reflect off of surfaces, and that reflection then bounces into our eye, and that's what's reaching our eye and is being registered by our rods and our cones, our photoreceptors in our eye. So let's actually talk about the pathways that light has to take to get to those photoreceptors, to those rods and cones. So not only is light bouncing off of some surface before it reaches your eye, when it actually enters your eye, there are kind of quite a few components of the eye itself that the light has to travel through in order to get to the photoreceptors. So the retina specifically, that is the layer that contains our rods and cones, that contains our photoreceptors. But to get there, light has to first travel through the air. Then it's going to travel through the cornea here. Then it travels through the aqueous humor. From there, it then hits the lens, which is here. So the anterior segment here contains the aqueous humor. The lens is here, this kind of big circular disc. Then it goes through the vitreous humor, which is found in the posterior segment, and then it finally hits the retina. And what's important is that every time there are three areas, the cornea, when it enters the lens, and when light leaves the lens, that light actually is bent. So when light travels through different media, light bends. And so because of that, there's a lot happening in like with the muscles of your eye and your lens itself that are working to make sure that light is actually hitting in the right spot of your retina and hitting those photoreceptors. So light does bend, but if it's focused properly, it's going to land on that retina, and that's when we're going to activate our photoreceptors. So let's actually look at how that works. So the photoreceptors themselves are actually organized kind of in a funky way. They almost look like they're backwards, which makes understanding how this activation pattern happens a little bit funky. So what I want to point out first is that notice that again, light is coming from, if we kind of look at our big picture here, light is entering the eyeball and it's traveling into ultimately hit the retina. And if we were to zoom in on the retina, which is being shown in the bottom portion of this figure, we would be able to see our rods and our cones and notice that they are actually kind of physically embedded in the retina. And then there's a bunch of stuff kind of sitting on top, which are the bipolar cell layer and the ganglion cell layer. So light actually travels past some cells. It travels past these ganglia and these bipolar cells in order to get to the rods and the cones. So I just kind of want to point that out, that there's this stuff on top, like kind of sitting on top of the rods and the cones. They're important, but they're not what's absorbing light. It's these guys here that are absorbing the light. And then eventually, basically what's going to happen is these rods and cones are going to be activated, and they're going to signal to the bipolar cells and the ganglion cells. So let's actually look at how the rods and cones are activated. We're going to specifically look at the cones, but the rods, but the same thing is happening for the cones as well. So I don't want you to memorize everything, all of the steps written in blue on this figure. I think it nicely shows what's happening to the photoreceptors when they are either inactivated or activated. So this specifically is showing what happens in the rods. Notice the rods get their name because they look kind of like a rod shape here. In the dark, what I want to highlight is just that we have these rods, and those in the dark, the rods themselves are actually constantly signaling to the signals in the brain. So photoreceptors here, they're going to talk to bipolar cells, which then send signals to a ganglion cell, which then sends signals to the brain. So this is kind of the signaling pathway that happens in order for us to see. What's kind of interesting is that in the dark, these rods are activated, and they are going to be releasing a signal here to the bipolar cell. And when that bipolar cell is activated, it's actually inhibiting this ganglion cell. So in the dark, the ganglion cell, kind of the last pathway to the brain, is inhibited. So that means that we're going to send no signal to the brain. So in the dark, we're telling the brain there's nothing to see, so no sight in the dark. Here's what happens in the light, though, that's being shown on the right. When light strikes our photoreceptors, that actually tells this signaling between the rod and the bipolar cell to stop. And so now this bipolar cell is getting a different signal. It's now activating. So now it activates our ganglion cell. So in the light, our ganglion cell is active. and it's going to send a signal to the brain that says, I'm seeing something. I have sight. So in the dark, we are basically turning off this final ganglion cell. When light strikes our photoreceptors, we change that communication pathway so that this final ganglion cell tells our brain that we are seeing something. So rods and cones are just going to be, they're both activated in the same way. In the dark, they inhibit that ganglion. In the light, they are activating that ganglion cell and telling the brain that we either don't see or we see something. But what the type of light that rods and cones react to is very different. So the table I've put here, cones, rods I want to point out are typically for our non-color vision. So think like at night, if you ever wake up in the middle of the night, you can see shapes and shadows and it's kind of a black and white color. Those are your rods being activated. In bright light, your cones are activated, and there are cones for different colors, but together when they are activated they mix, which is why we can see kind of a whole color spectrum. So think black and white for rods, color for cones. Rods have very high sensitivity. This is why they are going to be functioning best in dim light, whereas cones have low sensitivity. They function best in bright light. We have kind of this low acuity versus high acuity, not super important for this class. Other things I want to point out are that we have a lot more rods than we do cones. So we have greater number of rods versus cones, and where we find these guys in the retina is also different. Mostly cones are on the periphery. If you've ever kind of paid attention, if you kind of put your hand in front of your face and you move it towards the left or the right, you might notice that you lose color sharpness of your hand and it looks more black and white. It's because you're hitting your kind of periphery of your retina where the rods are more numerous. So their location is different. And this diagram from your textbook here really shows you just kind of a nice comparison between the rods and the cones. Again, rods are rod shaped, whereas cones are cone shaped, which is where they get their name. And then you can see an actual electron micrograph, so a microscope picture of what those structures look like as well. So this diagram is just kind of cartoon versioning the actual thing here on the right. Okay, so again, rods and cones are both going to be activated in either dim or bright light, depending on which photoreceptor we're talking about. And then just again to highlight now what's going to happen, how do we get to the brain to tell our brain that we are seeing? Again, our rods and our cones will be activated, and that way they're going to send a signal to the bipolar cells. So the bipolar cells are a type of neuron. They're a bipolar neuron. And those bipolar neurons then signal to the ganglion cells, which are another type of neuron. Those ganglion cells, they leave the eye. So if you kind of look back at the kind of overview of the eye, those ganglion cells are going to leave through the optic nerve at the blind spot. So those ganglion cells leave, and that's what's going to go to our brain, which is the third step. So those ganglion cells, they're either inactive in the dark or active in the light. They're going to send a signal. And specifically, that signal leaves the eyes. So in both cases, we can see that we create this optic nerve. So we have the optic nerve for the right and left eye. And those optic nerves travel to the visual cortex. And those ganglion cells then, so they're essentially sending their signal all the way to the visual cortex. And it's here that our brain can then think about and look at and perceive what we are looking at. So the visual cortex of the brain is where that information goes and is basically computed. so that we can see. All right, so those are the kind of quick basics of light. The other senses are fairly straightforward, so you should be able to do just fine by using those study guides and those review guides for the chapter. But again, if you have any questions, reach out.